As I mentioned previously, development has fascinated scientists for centuries. And it was in fact Aristotle who performed the first studies on comparative embryology. And he coined many of the terms that we still use in developmental biology, including oviparity, viviparity, and ovoviviparity. Oviparity refers to animals that lay eggs which hatch outside of the body, like birds. Viviparity refers to placental development followed by a live birth, as seen in mammals. And ovoviviparity refers to animals that develop in eggs, but these eggs hatch inside the body, as seen in sharks. Aristotle also coined other really important terms which we'll discuss later. But other observations that he carried out led him to support the idea of epigenesis, which states that an organism forms anew with each generation. So in other words, all of the organs and structures of an organism are made from scratch at each generation. And this idea is correct, and to us, it seems obvious. However, at the time, there was another predominant view known as preformation. And those who supported preformation incorrectly postulated that organisms are preformed within the sperm or egg. So they believed that the organism was already fully formed in miniature form within one of the germ cells. Although the idea of preformation may seem ridiculous to us, this idea was actually supported by many until the early to mid 1800s. Some scientists like Marcello Malpighi, who lived in the 1700s, made observations that led them to believe that organisms were preformed. When he observed what he thought were unincubated chick embryos, he saw a lot of detail. For example, he could see that the heart and the vessels were already apparent. Based on that, Malpighi concluded that the embryo had to be present in the germ cells, fully formed, and that development only involved growth of structures that were actually already present. However, what he didn't realize is that these eggs were not truly unincubated because they were left out sitting in the sun during the summer. So there was already development happening. Preformation was also an attractive idea to some scientists because if all the structures were already present at fertilization, then no external force would be required to guide development. So this seemed like the most simple explanation. In addition to this, at the time when preformation was a popular idea, the tenets of cell theory had not been postulated. So in other words, these scientists did not know at the time that the cell was the basic unit of life. They also lacked an understanding of the limitations to cell size. So they didn't realize that cells can only grow up to a certain point because of things like the rate of diffusion of molecules and the need to maintain an adequate surface area to volume ratio for absorbing nutrients and releasing waste. Evidence against preformation and in support of epigenesis came from the field of genetics. For example, children of mixed-race couples usually have an intermediate skin color relative to the skin color of their parents, and that pattern of inheritance would not be possible if development was only dictated by either the sperm or the egg. In other words, if children were preformed in the sperm or the egg, they would either have the skin color of their father or the skin color of their mother. Other evidence for epigenesis came from more careful observations by other scientists who show that there are some structures that are only present during development and are absent later in life. One of these structures is the notochord, which is a rod of mesoderm that is present in embryonic stages, but is lost during development. It's shown here in red in the zebrafish and in pink in the mouse embryo. This is the head and this is the tail. So the notochord is present there during development, and as I mentioned previously, and we'll see later in the semester, it's important for inducing the ectoderm that's above it to form the neural tube. However, this structure is not present in the adult, though I should mention that the remnants or the leftovers of the notochord contribute to the nucleus pulposus of the intervertebral discs. Despite this, the fact that the notochord as a structure does not have a counterpart in the adult suggests that development happens anew within each generation. Given that the presence of a miniature man or a miniature woman within the sperm or egg is obviously an incorrect idea, how do we explain the processes that drive development? If epigenesis is true and all organs form from scratch in each new organism, what is the force that directs the formation of these organs? Well, a German philosopher, Immanuel Kant, and a biologist, Friedrich Blumenbach, postulated that there is indeed a force, which they called developmental force, that guides development. They also said that this force can be observed and that it is present within the sperm and the egg. So there is something that is driving development, and that something is the instructions that are present in the genetic material of each organism, so our genes. So the modern view of developmental biologists is a mix of epigenesis and preformation. The instructions are preformed, they're present before fertilization, but the tissues and organs form anew.